When testing for allergens in your laboratory, you must consider how you can deliver fast and reliable results to your customers. The SENSISpec ELISA kits from Eurofins Technologies provide a quick and sensitive method for allergen detection. It's common for labs to receive many samples at one time, requiring a simple, streamlined process to manage and analyze these daily samples. It's critical to ensure that low-level contamination is not overlooked during your analysis to avoid reporting false negative results. With our SENSISpec Allergen ELISA kits, you can quantitatively detect the smallest traces of allergens in the sample in just over an hour. A broad portfolio of allergen ELISA kits is available to meet all your needs in the lab. Which allergen are you looking for? Whether it's proteins from milk, egg, tree nuts, seafood, or other food types, our SENSISpec Allergen ELISA kits will detect them all from a single extract. How to analyze for allergens? You will find all the reagents you need in the box. Simply weigh out your samples and add the extraction buffer. Place samples in a 60-degree water bath for 15 minutes, shaking every 2 minutes. This final extract can now be analyzed with all the SENSISpec Allergen ELISA kits you need. Pipette sample extracts and standard solutions into the micro-titer wells and incubate for 20 minutes at room temperature. Wash the wells 3 times with the supplied washing buffer. Tap the plate until the wells are dry. Add conjugate solution into the wells and incubate for 20 minutes at room temperature. Repeat the washing step and add the substrate solution into the reaction wells. Put the plate in a dark environment for 20 minutes to allow for color to develop. Add the stop solution to stop the color reaction. Read the quantitative results at 450 nanometers with an ELISA reader. Interested in learning more? We would love to hear from you. Contact Eurofins Technologies today.